Hi, my name is Alex Swarbrick. I'm a lab head from the Garvan Institute of Medical Research in Sydney, and it's my absolute pleasure to be moderating this session on spatial biology. I want to start by giving you an overview of the area and some of the kind of justifications for why we need to understand spatial biology, and then some of the technologies that are coming through that are enabling research in this area. So, I think it's well accepted now that solid tumors are ecosystems of cells. They are not simply homogeneous collections of neoplastic cells. We also know that there's great heterogeneity within the tumor microenvironment, both within the cancer cells themselves, but within the many parenchymal and host cells that reside within that tumor. And we know that those cells are not simply passengers, but play really central roles in determining the etiology of disease, right from its earliest stages through to governing treatment response, metastatic dissemination, and so on. Furthermore, there's increasing evidence that these cells are not simply jumbled together or admixed within the tumor mass, rather that there is structure to this. So, for instance, distance from a blood vessel determines the oxygen tension within a tumor, and so that sets up gradients of hypoxia and therefore gradients of gene expression. Growth factors and morphogens diffuse over distances, again, setting up gradients. And so I think while we need to understand the cell types that comprise tumors, we also need to understand exactly how they're arranged. And then, of course, there are other factors providing spatial context and cues to the tumor. For instance, the extracellular matrix, which again can define domains and borders within tumors that are almost certainly of significance biologically. So I think a great example of the kind of structures that we find within solid tumors are tertiary lymphoid structures. And so these are essentially aggregates of immune cells that resemble lymphoid structures like lymph nodes, in that they are immune cells coalescing in an organized fashion. And so typically we have B cells in the center here surrounded by T cells and a number of other cell types like stromal cells and lymphatic endothelial vessels. And we've observed these in solid tumors for many years, but the significance of them to solid tumor biology is still really not very well understood. But I think speaking to the potential importance of structure and morphology within tumors, a number of recent papers showed that the presence of tertiary lymphoid structures predicts response to immunotherapy, for instance. So what I'm showing you here is a figure from a recent paper showing that the patients with the highest number of tertiary lymphoid structures, so the red trace here, had a substantially better survival and response to immunotherapy than other patients. So arguing that there is a really major functional significance to understanding the organization of cells within a tumor. So how do we start to understand this then? We've used methods for a very long time that can provide us with beautiful spatial resolution. So H&E staining of a paraffin-embedded section here on the left, for instance, shows you this exquisite spatial organization and our ability to measure that. But of course, this is extremely low data content here. We're essentially measuring two parameters. We can use immunofluorescence for say 3 or 4 parameters, but still this is a very shallow look at the actual biology of the cells. Now on the right, in comparison, new methods like single-cell RNA sequencing are providing us massive data density per cell, but we completely lose the context. We lose the understanding of how those cells sit in space. So the challenge then is really, how do we put gene expression in its place? How can we combine the best of systems that allow us to go very deep into cellular phenotype and gene expression and genomics and epigenomics and so on, and put that into spatial context? Well, lucky for us, there are a number of solutions emerging now in recent years to this problem. And I've put examples of, I guess, 4 of the most common technologies here. The first one is based on, I guess, a quantitative extension from existing methodologies, so multi-parameter immunofluorescence. So in the top left here, we have a section stained with multiple antibodies on the vector system. There are other manifestations of this technology like Codex that uses barcoded antibodies, allowing you to increase the multiplicity of antibody density. In the lower left is an example from the ReadCore platform, which is a commercialization of the FISSEQ technology. This and other technologies like SeqFish essentially do in situ sequencing. And so this is a really interesting, novel solution to the problem. In the top right, we have an example of some data from the 10x Genomics Visium platform. And so this is essentially an array of capture probes across a slide, and it gives us an array of essentially transcriptomes. And in the bottom right, an example of kind of a cartoon demonstrating how we might use the Nanostring GeoMX platform, which allows us to do targeted measurements. So these are four really kind of different approaches to the same problem. And I think the reason we need so many solutions is this is a complex space. So as you can imagine, in spatial analysis of a tumor, we're trying to solve multiple problems. Ultimately, we need high resolution. We need massive multiplexing. We want to be able to measure many, many proteins, or the whole transcriptome, for instance. We need it to be sensitive. So we need to be able to measure not just highly abundant molecules, but also molecules that are important to the biology of cells, like transcription factors. And finally, we need these technologies to work on the kind of tissues that we typically have access to. And certainly in translational research, that tissue is often formalin fixed, paraffin embedded. And so none of these platforms really solve every one of these solutions. And so I think that's why we're very fortunate to have multiple solutions now emerging. I'm going to spend a moment just introducing the Nanostring platform, the GeoMX DSP, which I think is a really fantastic solution to this problem. And it works a little differently to other platforms. So and given that we have a number of presentations featuring it today, I thought I would briefly introduce it. So here I'm showing you a cartoon depicting the way that the transcriptome assays work. So on the left here, you can see we have essentially a hybridization probe binding to a target mRNA. And this probe is conjugated to a barcoding oligo via a UV cleavable linker. So when you hybridize these probes to your tissue, they bind to the tissue and to the mRNAs within it. We can then use UV illumination to break the UV linker in areas of interest and then isolate those barcodes and measure them. So this shows you roughly the workflow here. Starting at number one, we stain slides not only with a library of these oligos, but also with antibodies that allow us to get an immunofluorescent image to guide where we do our studies. We image the slide, select regions of interest. We then illuminate those regions of interest. Those barcodes are released, they're aspirated, and then measured. And in the newest kits, these measurements are done through next generation sequencing. And of course, as many of you will heard from Carl June's presentation, this technology can also be used by conjugating barcodes to antibodies. So with that, I'd like to finish my introduction, welcome you all to this session on spatial biology, and I look forward to 5 really fascinating presentations.
Dr. Alex Swarbrick is an associate professor of medicine at University of New South Wales, Sydney, NHMRC senior research fellow. He's also the co-head of the breast translational oncology program in the King Home Cancer Centre, Sydney, Australia. Alex will moderate the spatial biology track, where he will present his latest work on spatial expression in breast cancer and moderate the following speakers. First will be Arutha Kulasinghe from Queensland University of Technology. Next will be William Wang from the Broad Institute in Massachusetts General Hospital. Then we'll have Anuksha Dixit from Advanced Cellular Diagnostics. And then lastly, we'll have Kit Furman from Nanostring Technologies.
So I'm really excited to present to you some data from my lab today. This is all unpublished data, and using a combination of technologies to try and understand the tumor microenvironment of triple-negative breast cancer. So first, my disclosures. Okay, so I've already described to you in my introduction how we think of tumors as ecosystems. And so the approach that we take in my lab to try to tackle this problem and understand the way these ecosystems function is several fold. The first is really that we need to understand the taxonomy of this ecosystem, this cellular environment. Even though we have great histological and morphological definitions of cells, we have pretty shallow molecular definitions of cells. And so really our first challenge is to understand the cells that are present, which of them are important, what are the gene expression programs, the signaling pathways, and so on that govern their behavior. And finally, what's their expression of therapeutic targets? Because we need to know that if we aim to change their behavior, intervene. We then need to understand how these cells are organized in space, the way that their organization influences their phenotype, and then ultimately to try to develop novel treatment strategies based on this information. I'm going to talk today quite a bit about tumor-infiltrating T cells in triple-negative breast cancer. Now, the reason is that of all breast cancer molecular subtypes, triple-negative breast cancer has a very poor prognosis. We still lack a good collection of targeted therapeutics for triple-negative breast cancer. In this disease, T cell density is prognostic. And in fact, a proportion of women with triple-negative breast cancer do respond to immune checkpoint therapy, though this is a relatively small proportion, and interestingly, only in the context of neoadjuvant treatment, really, which is where treatment is given into women with a localized tumor in their breast rather than metastatic disease. So we really want to understand more about the way anti-cancer immunity works in triple-negative breast cancer. There's a bit of signal here that suggests to us that there's more to find. And in trying to understand the way anti-tumor immunity works in many solid cancers, often the model that people use to think about is one where we compare patients. So we look at intra-patient heterogeneity. So in other words, is this tumor hot? Is it cold? Is it an immune desert, for example? But we want to understand more about the way that immunity works within a tumor. So for instance, do T cells exhibit intratumoral heterogeneity? Is it enough to just characterize a patient with a single measure, or do we need to really be looking in different domains across the tumor? Is that phenotype of T cells influenced by their local tissue context? And then could we potentially use this information to develop more effective immunotherapies? So to approach this problem, the first place we've started is to develop a cellular atlas of early breast cancer. And here we're using a very large collection of viably cryopreserved breast cancers that we've been collecting for a number of years across all the subtypes of disease. These tissues are dissociated, and every cell that we're able to recover undergoes single-cell RNA sequencing, as well as a suite of other methods, including CITE-Seq, which I can elaborate more on in question time if people are interested. And what this gives us then is a really wonderful taxonomy of the cells that are present within a breast tumor, acknowledging, of course, that not every technology captures every cell. So this is a very high-level view of the first snapshot of about 30 cases here from this early breast cancer cellular atlas. And you can see that many of the cell types we know to be present in breast cancer are represented here. And when we conduct really detailed annotation of these cells, we break this down into a really detailed taxonomy. And you can see here the major classes of epithelial, immune, and stromal cells breaking down into subsets of cell types and ultimately into states from these cells. So I don't have time to talk about the stromal or epithelial populations today, but I want to talk briefly about the T cell data as it pertains to spatial profiling. And so here I'm showing you a UMAP clustering of all the T cells from our breast cancer atlas here. You can see that there are many diverse phenotypes present. This also includes some innate lymphoid cells, and NK cells, and so on. But what's really interesting is that we see a really wide diversity of phenotype of T cells present within breast cancers and in triple-negative breast cancers. Most of these states, interestingly, exist in every patient. So it's not like one patient has type A and another patient has type B. Most of these states are existing in every one of the tumors that we observe. And while all of these states can be detected in every subtype, interestingly, there are 3 of them that are enriched in triple-negative breast cancer, and I've highlighted them here on the right. The first one is a cell type with a strong activated interferon signature, which is associated with effector T cells. We have a proliferating population here, cluster 11, marked by Ki67 and many other proliferation markers that's enriched in triple-negative breast cancers. And finally, a population in cluster 8 here with many markers of T-cell exhaustion. And it's interesting because these 3 populations are really the populations that we would associate with a tumor that's got some kind of anti-cancer T-cell activity. So again, arguing or starting to explain perhaps why triple-negative breast cancer has some response to immunotherapy. So the challenge then is, how do we put this in its place? How do we take this data and start to understand how much of this diverse phenotype of T cells within every tumor is actually related to their location? And so we've been lucky enough to collaborate with Nanostring on a project piloting a really exciting new technology, which is their whole transcriptome assay. And so this experiment has been a little bit interrupted by COVID-19, but so far we've managed to collect data from 6 cases. So these are triple-negative breast cancer FFPE blocks we've cut. These are between 2 and 5 years old. We've then masked them with 3 different masks to identify cancer cells, T cells, and stromal populations. And then we've used this ability of the GeoMX to choose regions of interest, and we've selected 24 areas of illumination where we've used the UV light to capture gene expression. And these samples have been hybridized with a whole transcriptome panel, so 18,000 probes against the very large majority of the genes that we want to be able to measure. So really very exciting expansion in the multiplexing capability of this platform, all on paraffin. So this is the experimental design we're using. We're interested very much, as I mentioned, in T cells and in understanding the way that T cells microenvironment affects their biology. So by using a combination of the composite immunofluorescence and the H&E stain, we're able to select T cells residing in the stroma, nowhere near cancer cells. We can find T cells in tertiary lymphoid structures. We can capture either them on their own or the entire tertiary lymphoid structure. And we can also capture T cells adjacent to cancer, which interestingly we find in every one of these cases, there are always what appear to be pockets of resistance. So T cells accumulating next to cancer cells. And so using the masking, we're able to collect not only the CD3 positive fractions to get the T cells, we can also capture the adjacent cancer cells or stromal cells and so on. So looking now at our data from this is now all generated through next generation sequencing of these libraries. So a very high level view of the sensitivity. Of course, you might be concerned that we're working with paraffin here, and you kind of wonder what kind of sensitivity we're able to achieve. First, I'm just showing you here the kind of areas of illumination that we've collected. This is the area here on the x-axis, and on the y-axis is the number of nuclei. So there's a very nice correlation between area of illumination and the nuclei contained within it. As you can see, of course, that different cell types have different sizes and cell densities, so they form, kind of different correlations here. But what's really pleasing to us is that we are detecting a really substantial number of genes in each of these areas of illumination, a median of something like 7,500 genes per AOI, which is really very impressive, I think. As you can see, as you'd expect, that there's a relationship between the number of nuclei that we capture and the number of genes that we detect in each of those areas of illumination. So now using unsupervised clustering, this RNA-Seq data, well, sorry, not RNA-Seq data, but gene expression data from each of these areas of illumination, this naturally falls into blocks of gene expression here, which is really nice. The first track in the legend on the heat map here is the type of area of illumination. You can see that the cancer cells in green, they all cluster together. The primarily T cell and TLS derived populations cluster together, and then the stromal cells cluster together. So that's nice. The expected biology falls out here. And then we have kind of patients stratified within it. Interestingly, the host cells within patients are often very similar in their gene expression features, whereas the cancer cell gene expression features are unique to every patient. And you can see that in the blockiness of that top left part of the heat map on the left. And this is something we observed with single cell RNA sequencing as well, that every patient's cancer cells are profoundly different in their transcriptome, whereas their immune and stromal cells are very similar. On the right, you can see just a different way of visualizing the data using a t-SNE projection here, that you can see that generally the host cells cluster down in the bottom left, the cancer cells cluster in the bottom right, and again, you can see every patient is different here. Now, one of the really nice things about this data is that we can, while we know that every measurement we're taking is a collection of cells, this is not single cells, obviously, we can use methods like deconvolution to start to understand the contribution of different cell types to each of these areas of illumination. And this is an area that Nanostring have been working in a lot for a very long time, using their bulk gene expression assays, and now they can apply this to the GeoMX system. And so what we can do now is use published signatures of endothelial cells, fibroblasts, B cells, and so on, to predict the proportion of cell types in each of these AOIs. And so again, you can see now that the cancer cell regions where we thought we were sampling cancer cells are predicted to be composed almost entirely of cancer cells, stromal regions of fibroblasts and so on, T cell regions primarily T cells and so on. But we do also see some interesting observations like B cells present even within some of these T cell enriched regions, which I think is something interesting that we want to follow up. But I think what's really powerful now is that because we have matching single-cell RNA sequencing data from each of these cases, we're able to use our high-resolution taxonomy that I showed you earlier and ask, can we use this to deconvolute cellular proportions present in each of these AOIs? And we can. And so you can see here some of these many populations that we're able to measure within breast cancers using single-cell RNA sequencing data can be detected very robustly within the GeoMX data. And so this is really nice. We can start to look for relationships between cells, and you can already see obviously relationships emerging. For example, at the top there, we see CD8 T cells, memory B cells, and naive B cells clustering very nicely. And this is probably in part driven by their co-occurrence within tertiary lymphoid structures, as well as in other places. Now, this data is a little bit dense to look at, so I've pulled out some examples here that start to address more specifically the questions we had around T cell phenotype. So first, just to show you that deconvolution returns expected results with our own single cell data. So these data sets appear to be compatible. Our fibroblast signatures are heavily enriched in the stromal populations and depleted in all the other areas of illumination. Similarly, endothelial cells, which we did not target, are across any of these areas of interest. So this is a positive control and a negative control, essentially. When we start to look at T cells, we can see some expected results here. So CD8 T cells are enriched at the interface with cancer and are depleted in either the stroma or in tertiary lymphoid structures. So this might have been what we expected, that the cells interacting with cancer cells proliferating are primarily CD8 T cells, which is interesting, whereas CD4 T cells are depleted at the cancer interface but are enriched at tertiary lymphoid structures. And we certainly know that to be the case from the literature. Looking now more deeply at these subsets of CD8 T cells, which is really what we want to try to understand, I pointed out to you some of these populations earlier in our single cell data. And what's really cool is that we can now show that it's at the cancer interface that we get enrichment for these interferon positive CD8 T cells, for these cycling T cells, and so on. So again, letting us link populations identified from our single cell data and showing that these cells are actually the ones in contact with cancer cells. Furthermore, looking at details of the gene expression features of these AOIs gives us further insights into the state of these T cells. So when we look at gene expression features now of the T cells next to cancer in green, or the T cells in the stroma or TLS structures, the purple and yellow tracks on the left there, you can see that the T cells adjacent to cancer have markers of maturation, so transcription factors like ID2, RUNX1. We're able to measure these quite robustly, and these are arguing for T cell maturation. We can measure high levels of the actual molecules that CD8 T cells use to kill, so granzyme and perforin, for instance, and then obviously molecules associated with activated interferon signaling like IRF1. So kind of stepping back now, what this really suggests to us is that subsets of T cells adjacent to tumor cells are proliferative and have all the signs that we might expect of activated T cells. So elevated interferon signatures, proliferative signatures, cytotoxic signatures. And what's interesting is that we find these in every case. So despite these tumors growing, these women presenting with disease, there appear to be what you could call pockets of resistance, pockets of T cell activation occurring in these tumors, but perhaps these are too infrequent to sustain sufficient anti-tumor activity. And so really our thinking here is that if we can understand more about these pockets of resistance, we might understand how we can make them expand in number and in scale. Okay, I want to finish up by showing you another vignette, really, and this is a single case, I think, but just demonstrates some of our thinking when we start to try to apply this technology to understanding neoplastic cell heterogeneity, so the cancer cells themselves. And the reason we're interested in this, of course, is we know that cancer heterogeneity is a poor prognostic factor in many solid cancers. Certainly, we observe this quite frequently in our single-cell data sets. What I'm showing you on the left here is single-cell data from a triple-negative breast cancer, and each of the populations that I've circled are actually cancer populations based on marker gene expression and other measures, as you can see on the right. Yet there's clearly quite substantial gene expression heterogeneity between these populations. So we are really interested to understand how much of this is driven by genomic differences between those populations, how much of that is driven by essentially the location of those populations. So I'm going to skip over this slide in the interest of time. And so the experiment here is to take one of these cases, and interestingly, we often see in cases with large heterogeneity at the gene expression level, we also find morphological heterogeneity. So here's an interesting case where we see on these H&E stains here, multiple different morphologies as identified by our anatomical pathologist. And so we sampled these different morphologies using the GeoMX DSP and looked at gene expression features here. And what I'm showing you on the right is a heat map of the largest gene expression differences between each of these regions. And you can see blocks of gene expression that are unique and distinct to each of these morphologies, which is really interesting. And on the left, I'm showing you that these different morphological regions cluster based on their histomorphology. So this is quite interesting that morphologies associate with gene expression features. Digging into just a few kind of signatures here relevant to the earlier part of my presentation, we can see, for instance, the different regions have altered gene expression in pathways relating to T cell recognition. So, for example, MHC class 1 expression is very high in this orange cluster here. And interestingly, this population actually resides at the edge of the tumor right adjacent to some of those T cells that we were talking about earlier. Similarly, this population also has high interferon signaling, linking it with the observation we made in T cells earlier. We can also see in different populations, for example, that evidence for strong activation of ERK signaling and AKT PI3 kinase signaling. So this is now starting to allow us to get evidence that indeed some of this heterogeneity is driven by spatial organization of these cells. And there are some really great tools that we have available to us now through this system that allow you to start to map morphology onto tissue and then layer gene expression features on top of that. And I won't go into detail here, but these are really great tools that allow us to visualize some of our data to make this very complex multidimensional data more understandable. So I'll just finish up by summarizing some of the findings here. And we're developing this cellular atlas of localized breast cancer. We're also working on prostate cancer and other diseases. And using that, we've found a number of novel cell types and states that I didn't have time to talk about today. But what we're really pleased about is that this data seems to be compatible with the GeoMX DSP, which can be used not only for discovery work, because this is a whole transcriptome solution we're using here, but also to integrate with our single cell data. And so these methods like deconvolution methods can allow us to start to drill down into the details of the cell types residing within each of those areas of illumination. Using differential gene expression analysis, we've shown that some of these T-cell phenotypes that we see are divergent based on their location within a tumor. And of course, we need to conduct these studies in much larger cohorts of disease to strengthen these findings and then to validate them using other methodologies. So I just want to finish up by acknowledging the many people who've contributed to this work. This is a really large body of work, both the single cell assays as well as the Nanostring GeoMX work here, in particular Kate, Sandra, Dan, and Tony, who've been instrumental in the Nanostring DSP analysis. We worked with a number of wonderful scientists at Nanostring, and I really want to acknowledge their contributions. Our clinical collaborators, of course, give us not only access to tissues, but Sandra O'Toole here is also a really fantastic breast specialist pathologist and has been very important in helping us do the spatial studies. Finally, I'd like to acknowledge my funding agencies, the National Breast Cancer Foundation and the National Health and Medical Research Council. And if you're interested in our work, I'd love to connect with you. Please find me on Twitter at Dr. Alex Swarbrick. Thank you.
Thank you, Alex Swarbrick, for that great presentation. We're going to follow up with a few questions. The first question I have for you is, how do you annotate cell types with such confidence in your single cell data?
So that's an important question, namely, how do we get confidence in annotating different cell types within our single cell data? We go about this in a number of ways. Firstly, of course, we use marker genes, and so there's obviously an established literature on many of the marker genes for different populations of cell types. However, some of our work has revealed deficiencies in that. One of the other tools that I didn't really have time to talk about in my presentation that we use a lot is CITE-Seq. And so I'm showing you on the slide now just a little cartoon overview of the way CITE-Seq works. So here we have barcoded antibodies that are, and those barcodes are polyadenylated. So this is in some ways kind of similar to the nanostring barcoded antibody solution, except that we're staining cells in suspension here rather than on tissue. And so these barcoded antibodies bind to cells, they get encapsulated, the polyadenylated barcodes get captured along with cellular mRNAs and end up in your sequencing library. And so like flow cytometry, then you're measuring the abundance of a molecule on the surface of the cell, but instead of using fluorescence, you're using sequencing of a barcode. And there are a lot of advantages to using CITE-Seq here. Firstly, because it's a FACS-like technology, it allows us to link back to FACS-based immunology. And so when we're trying to annotate particularly immune cell populations, CITE-Seq has been really very valuable in giving us confidence that the cell type that we think it is from the RNA sequencing data also has cell surface markers that are compatible with the existing knowledge. The other nice thing about CITE-Seq is it's both sensitive, but also can be massively multiplexed. And so we're using libraries of somewhere around 150 antibodies currently here because they all just have a different barcode. So that's quite easy to use. And finally, of course, this is really important to us when we consider therapeutic targeting of proteins. Finally, the other method that we use in annotating cell types is in signatures and transcriptional signatures. And so obviously when you have single cell RNA sequencing data, you can interrogate that data very deeply, and we can predict the transcriptional drivers of different cell populations. So in some scenarios, that's also really valuable in us helping to identify distinct cell types.
And how did you think about choosing the areas of illumination on the GeoMX platform on your samples?
So perhaps unlike other technologies, choosing the areas of illumination is really one of the key rate limiting or critical points in the GeoMX workflow here. Really, you're whether your data addresses your question comes down to how effectively you can identify and label those areas of illumination. And so this comes down to a number of things. I think probably primarily the first is that you really have to have an understanding of the tissue that you're working with. You really, we probably spend a couple of hours looking at every case that we want to study and really thinking about the regions within that tissue that we're interested in. We work very closely with an anatomical pathologist that helps us mark up that tissue and so on. So I think that's really the first thing that people need to understand. You don't simply take a piece of tissue, throw it on the platform, and expect to get good data. The second really comes down to how well your masking works. And so this is really based on your choice of antibodies that you use to label different cell populations of interest. So you need to both design a strategy that allows you to cleanly identify the populations you want, and you've got to make sure those are quality reagents that you're able to use as well.
When would you choose to use GeoMX over other technologies for your research?
So this question of which spatial technology to choose is obviously a really interesting one. There's no one answer for everybody's application. Different technologies really suit different spaces. The GeoMX, in my experience, has a number of really valuable applications. I think the one that we're really excited about is applying it to human specimens that are either very small, very precious, or poor quality, for example, but which have really important, rich clinical annotations. So, for instance, response to therapy data or survival data associated with them. Many of these cohorts are in paraffin. If these are metastatic cohorts, often you have very small pieces of tissue that might not have been very well sampled, and it's going to be very difficult to work with them with most technologies. What we're excited about is I think the GeoMX is going to be really great in that setting, at least, in understanding molecular features of these very valuable clinical specimens.
And in your experience, what are the smallest areas of illuminations that you can use to ask meaningful questions?
So this question of how small your areas of illumination can be is obviously one that's everyone's interested to understand. Look, we ourselves haven't done any benchmarking of this. I know that Nanostring have, and there's probably some published data on that. What I can show you, though, is one of the panels that I showed earlier in my presentation, which plots essentially number of genes detected versus number of nuclei. And so you can see that obviously there's a relationship between the number of nuclei and the number of genes detected. But what's really quite surprising is that in certain areas of illumination, at least, we're detecting above median, so almost 8,000 genes in a sample that was probably something like 20 or 30 cells. And obviously this is going to depend upon the transcriptional activity of the cell type that you're looking at, and other considerations like the density of the cells and so on. But I can certainly say we're quite pleased by this, and this is an experiment we plan to do, which is essentially a titration, looking at across different cell types, what kind of sensitivity can we achieve based on different areas of illumination.